Penicillins remain the drug of choice for a significant number of infectious diseases. The structure of penicillins are basically thiazolidine ring connected to a beta-lactam ring or citric amide ring. Then beta-lactam ring is also attached to a side chain. So penicillin is obtained by natural process using fermentation, and it was discovered by Alexander Fleming in the 1940s. So penicillin nucleus itself is the chief structural requirement for the biological activity. In side chains can be added. So for penicillin there are two disadvantages. It's broken in the acid, in the presence of acid, or as well it is being inactivated by beta-lactamase enzyme that are found in the periplasmic membrane space of the bacteria. Following oral absorption, penicillins are distributed widely throughout the body, and the antimicrobial spectrum of penicillin G, penicillin V, they are similar for aerobic gram-positive bacteria. Most streptococci remain susceptible. Penicillin resistant streptococci and streptococcus pneumonia are also becoming more common. Penicillin resistance pneumococci are especially common in pediatric population. So this is the structure of penam. Then we go to the next slide. So nomenclature of penicillin. So nomenclature of the penicillin as is complex. Chemical abstract system is definitive and unambiguous, but it is too complex also. Chemical name of benzyl penicillin sodium is monosodium 2S 5R 6R. So in penicillin we have three stereo centers, that is chiral carbon at 2S position, 5R position, 6R position, 3,3-dimethyl 7-oxo, 6,2-phenylacetamide, 4-thia, 1-aza bicyclo, 3,2-0-hept 2-carboxylate. So 3,2-0 structure comes with the joining here, that is the penam C5 and N1. So the left structure has 3 residues, 2, 3, 4 position, and left structure has 2 residues, 6 and 7 position. And between the bridge of C5 and N1 there is no other bridge, so it is named as 3.2.0. So if we don't have any substitution at the 6th position, this structure is known as 6-aminopenicillamic acid. So chemical trivial name for the added radical that complete the structures, so that is called carbamyl 6-aminopenicillinic acid. There are 3 asymmetric centers in the benzyl penicillin molecule as I have shown. So this is the mechanism of action of benzyl penicillin. So here you can see when we in the gram-positive bacteria, the cell wall is made of cross-linking between N-acetyl glucosamine and N-acetyl muramic acid. So here you can see the penicillin binding protein at the transpeptidase, it adds one unit of alanine. So slowly by simple cross-linking process, the penicillin cell wall structure is being formed. So when beta-lactam is coming, the structure of the beta-lactam, as you can see, mimics the structure of the alanine. So in that case, it competes with the penicillin binding protein site. So alanine is not bound for the cell wall preparation. In that case, the beta-lactam occupies the position and inhibits the cell wall synthesis. So molecular mode of action of beta-lactam antibiotic is selective and irreversible inhibition of enzyme processing the developing peptidoglycan layer. So you can see the structure here in NAG, that is the N-acetyl glucosamine, NM, and here we have amino acid linkage, alanine, glutamic acid, lysine, alanine. So it both 2 strands oppositely linked by a pentaglycine unit, and this structure is being generated in the common biosynthetic pathway, and penicillin inhibits the incorporation of alanine, that is the binding the cross-linkage site. So penicillin molecule contains 3 chiral carbon atom at C3, at C5, at C6. So we have two naming system, chemical abstract naming system and USP naming system. Accordingly, 2 position is equivalent to 3 position of chemical abstract system, and for the 4th position is 1 position. So for chemical abstract system, we start the nomenclature with sulfur, whereas with the USP system we start the nomenclature with nitrogen. If we see the structure, at the 2 position we have a carboxylic acid group, 3 position we have dimethyl group, and we have chiral carbon. Also 6th position we have an amide linkage, RCONH. The major problem with and if we are not having this amino group, this is called penicillinic acid. So you can see the structure. We generate different structural variants of the RCONH. The basic problem is penicillin, with that there is the breakage of the beta-lactam ring. So CO group is easily being attacked by the nucleophile, and the beta-lactam group is opened. So you can see this is the structure of penam, and this is the penicilloic acid. All naturally occurring microbiologically active synthetic, semi-synthetic penicillin have the same absolute configuration and have 3 centers. Carbon atom bearing acyl amino group C6 has the L configuration. Carbon 2 is the carboxyl group is attached, has the D configuration. So acyl amino and carboxyl group are trans to each other. All the natural penicillin are strongly drugs. Dextrorotatory, solubility and other physicochemical properties of penicillins are affected by the nature of the acyl side chain. So penicillin V, penicillin G, this is a natural penicillin, this is called benzyl penicillin. This is quite easy to break down by the stomach acid and environment, whereas we have generated, there are 2 different types of penicillin, that is called beta-lactamase sensitive penicillin, beta-lactamase resistant penicillin.